Now if we imagine the cell now, in metaphase 1, here's the cell. What happens is the tetrads all move towards the centre of the cell, like this. And they line up really quite neatly along the equatorial plane of the cell, along the middle plane of the cell. And of course they're in their pairs at this stage. Each homologous chromosome is in a pair. Like this. And this plane that they line up on here is called the metaphase plate. So they're lining up neatly on this metaphase plate. And remember that in either half of the cell there is this organelle called the centriole. And this is generating these meiotic spindles, like this, connecting to the cell, or connecting to the tetrad centromere. The bit in the middle was the centromere. So the centriole connects the centromere with these mitotic spindles made of microtubule protein, like this. So that's metaphase 1. Now the next stage is going to be anaphase. So things are now nice and tidy. It's all tidy on this in this central plate. But the tetrads are still coiled around each other. I mean, if we remind ourselves of where we left them before, we had centromeres there and there, and here we had one like this. So they're still coiled up, or still interlinked, like this. They're still all linked up. So this is the stage at metaphase 1. Now next we move into the anaphase. Anaphase 1. Now what happens here, I'll just illustrate this I think by looking at two, or maybe just look at one actually, or just look at one pair of chromosomes. So we've had these here in metaphase and we saw that they were crossed over. We had these crossover points where they cross over, the chiasma or the chiasmata, where they cross over. Now at anaphase, these chromosomes are pulled apart, or the tetrads are pulled apart. So there's one centromere there, there's another centromere there. And they're actually pulled apart by these microtubule meiotic spindles. They start to pull them apart. And when they pull them apart, what happens is, let me draw it. What happens is, that's pulled apart like that, so that's still got those arms. These diagrams are simplified. In practice, the arms are more interlinked than this. And here we have the bottom part of that. Now, what this tetrad does is it takes part of the other homologous chromosome with it. So it takes that bit of the chromosome there, goes with this half. So that green bit there links onto that red bit there. We can just draw that like that. That bit goes. And it's the same at the bottom. This bit and this bit stick together. So we have this bit here, and we have this bit here, now remaining together. So it would now look like this. So there's been mixing of the genetic material from the two homologous chromosomes. And it's the same here. When they separate, the crossover points connect, and there's recombination of the genetic material. So these tetrads are no longer the same as the parent tetrads were in the interphase. The crossover points stay, and these pull apart. So the homologous tetrads pull apart. They're pulled apart by these spindles, so that one's going to move that way. That one's going to move that way. But they've now got this recombined material from the different original tetrads. And this is so important because this means that there's now a recombination of this genetic material. The chromosomes are no longer identical to their parents. They've got a recombined genetic profile because of these essentially new tetrads that have been produced. And they pull apart now. So the homologous tetrads pull apart by these meiotic spindles. And they go into haploid halves. So now, as these move apart to that side of the cell and to that side of the cell, we're going to end up with 23 tetrads on that side of the cell. And we're going to end up with 23 tetrads on that side of the cell. So originally here we had the tetrads together, then they're going to pull apart, giving 23 in each half of the cell in this anaphase stage. The centromeres hold together the four sister chromatids still, but they're moving apart to have 23 tetrads in each half of the cell.